Okay, right-hand page. How does genetic information flow through a cell? Okay, so perhaps you're Catholic, or maybe you're not, and you just know what this word means. What does this word dogma mean? It's like the biggest idea it is. It's almost like a doctrine, okay? Here it is, the essential dogma of biology. Biggest idea in biology. DNA is transcribed into RNA, is translated into protein. Make sure you write that down. DNA to RNA to protein. Now here is the flow of genetic information. The process of going from DNA to RNA is called transcription. And the process of going from RNA to protein is called translation. Another part of our central dogma is that DNA needs to be copied. When in the cell cycle is DNA copied? You got 4 choices. Raise your hand if you think it's G1. S stands for synthesis phase. How confident are you in that hand raise? You should be very confident because it is in the S phase. Air 5. Okay, good job. DNA replication occurs in S phase. So before cell goes through division, it copies its DNA. Okay? So just a quick note, we'll go into more detail later, but RNA stands for ribonucleic acid. It's usually a single nucleotide chain, whereas DNA is a classic double helix, okay? Double nucleotide chain. It's like a twisted ladder. The DNA and RNA are both made of monomers called nucleotides, which we've gone over before. Tell your neighbor the 3 parts of a nucleotide. Okay. So there's 3 parts. I heard ATCG. Hey, that's a good start. Those are called the nitrogenous bases. And by the way, in DNA, A pairs with T, C pairs with G. That's called complementary base pairing. A pairs with T, C pairs with G. Okay? If you're ever stuck, here's how I quickly remember that. At spells a word, ta spells a word. It counts in Scrabble, ta. Okay? C and G, it's almost like you have to write the same letter, right? Because the G is just a C with a fancy 90-degree angle. If you're stuck and it just doesn't roll off your tongue like it does mine, oh, A pairs with T, C pairs with G. But I've been saying that for years now. Turns out that's just how it works, okay? So, in addition to having a nitrogenous base, there's also a sugar. The sugar in DNA is deoxyribose. The sugar in RNA is ribose. That's how they got their names. Okay? So it's just whatever sugar's there. And then there's a phosphate group. What's the chemical formula for phosphate? PO4, phosphate. The phosphate forms the backbone or the sides of the ladder, if you think of DNA being like a ladder, or the strand if it's RNA. Now this famous double helix shape was first discovered by Watson and Crick. Okay? I'm going to tell you how we copy or replicate this DNA. Replication of the DNA, tell your neighbor when it happens. I heard a lot of S phase. Good. Okay. I'm drawing a little twisty ladder of DNA. Right here, the phosphate groups are in the backbone. The nitrogenous bases are held together by hydrogen bonds in the middle. So this is like A and T. And then the sugar forms with the phosphate along the sides here. So the phosphate group is like the rungs of the ladder here. Okay? Right here, I just opened up this piece of DNA because that's what we need to do to replicate it. It's called a replication bubble. Okay? So let's see if you guys remember complementary base pairing. I'm going to make up a sequence of DNA. G, T, C, G, A, T. Okay? If this side of the DNA strand is G, what is this side? If this is T, this is? If this is C, this is? G pairs with? A pairs with? T pairs with? Ta-da. Okay? That's called complementary base pairing. One other thing about DNA is that it is anti-parallel. Like this. What that means is these, there are carbons in addition to the phosphate. They actually have numbers on them. You don't need to know where the numbers are, but we do need to know that DNA is replicated in the 5' to 3' direction. So if this strand of DNA is 3', the other strand is the 5'. Okay?